Okay, in today's lecture, we're going to continue the process of building towards making an organism. In the last few lectures, we've been talking about the chemistry of life. Now we're actually going to put something together, put components together, and make a living thing. So to start with, I'd like to review some of the stuff from last time. So the question here is, what type of chemical bond is represented in each of these figures, and which of the bonds is strongest and weakest? I'll give you a few seconds to think about that. You can also pause at this point. Okay, so if we go through these different bonds, A is a hydrogen bond, because you can see here's the hydrogen interacting with an oxygen, forming a link between the two. Bond B is ionic, because sodium has ionized into a positive cation. Chloride has ionized, picking up an extra electron to make an anion, and together they attract. The last of these is a covalent bond, in which the hydrogen ion or electron in the outer shell, which is right here and here, is being shared between two atoms. So this is a hydrogen atom, here's another hydrogen atom. This is combining together to form H2, and they form a covalent bond. In terms of strength, the hydrogen bond is the weakest of these three, and the covalent bond is the strongest. Second question, which class of molecules is represented by the chemical formula below, and explain your reasoning. And I think I actually left the reasoning already here. So I'm going to stop. No, I'll just leave it because this will take too long to go back and re-edit, but basically figure out what this is. And this is a carbohydrate. And the reason you know that is because for every carbon, there is one water molecule, so carbohydrate. Okay, so today's lecture is all about cytology. We're going to cover the cell size, the principles that limit or regulate cell size, talk about the basic components of a cell, and then finally go through some of the specific components. So cytology is the scientific study of cells, so it's basically cell biology. And so the first thing we're going to talk about is what limits cell size. Okay, so when we talk about cell size, we'll be using the metric system. And you can see here from this picture, basically everywhere that uses the metric system is in green, and everywhere that doesn't, namely right here. And for those of you that are from other parts of the country, Alaska and Hawaii, basically these are the regions that use it, as well as this state right here, country right here, which is Myanmar. So basically the US, Myanmar, and I guess Antarctica don't use the metric system. So when we talk about cell size, we are going to be talking about a micrometer, and a micrometer is 1/millionth, so 1/millionth of a meter, or a 1,000th of a millimeter. And just as a reference, if you look at a metric ruler, this would be your centimeter mark approximately, and then you've got the little hash marks in between. Those are millimeters. And so if you take a 1,000th of that, that is what we're going to be looking at when we talk about cell size. So the smallest object that is visible to the human eye is about 100 microns. So a micron is the same as a micrometer. And so if you look at that, look at something that we all normally look at, this is about 1/4 the size of a normal period at 12-point font in Times New Roman. So if we look at where we orient to size, the scale down here is showing sort of a range of sizes from smallest to largest on the left. This is a log scale, which we talked about when we talked about pH, so that basically every step, so if you go from 1 micron to 10 microns, there's 1 log, and so it's just turning this sort of, you multiply by 10 every time you go up. And so when we look at human cells, most human cells are going to be in this range of between about 1 micron and 10 microns. And you can see this is all material that you need a light microscope to see, which are the scopes that we use in lab. As you get smaller, we go into electron microscopes, and here's the limit of human vision. There are some cells that are much larger than that 10 micron size, normal size. And so if you look at a neuron, which we'll be looking at much more in depth later in the semester, neurons, the axon of a neuron can be up to a meter, sometimes over a meter long, as it goes from the spinal cord down to the bottom of our feet. Skeletal muscles can also be much longer. So muscle fibers that are in your thigh, running from basically your hip to your knee, can be up to 30 centimeters long. But it's important to remember that even though these cells are very long, they are very, very thin. So the cross section here is still going to be on the order of like 5 microns. And so it's like a very thin thread, and you can't see them without using a microscope. So what limits cell size? So cell size is limited by the surface to volume ratio. So there's going to be 2 ways to look at this. Here's the professional version using a cube. So you can see if you look at a small cube, each length of the small cube is 10 microns. And so to get the surface area, so surface area is this region. Again, you just multiply 10 by 10. To get 100, you multiply it by 6 because there's 6 sides to the cube to get 600 microns squared. If you go to the volume, you multiply one side by the other side by, so length, width, and depth. And so it's 10 by 10 by 10, or 1,000 meters cubed. So you can see there's a ratio of 600 to 1,000. If you double the size of length of one side, so it's now 20 microns, surface area is now going to be 20 by 20 times 6, or 2,400 microns squared. And if you go for the volume, it's going to be 20 by 20 by 20, or 8,000 microns cubed. And so you can see this is basically 1 to roughly 3. This is going to be 6/10, which is basically, it's a much smaller ratio, so it's greater than 1/2. So as you double the length of a side, you actually decrease the rate at which the volume is increasing is going faster. Another way to look at this that I like is looking at a sphere. So if you look at a sphere, just this is without actually putting in numbers. To calculate the surface area of a sphere is 4 pi r squared. So you've got this key. So surface area equals 4 pi r squared. Volume equals 4/3 pi r cubed. And so the key thing, this is a squared relationship. This is a cubic relationship. And so what this really means is as the cell grows, volume increases faster than surface area. And so since surface area is the region that is in contact with the environment, This is the part of a cell, if I need to spell while I'm not thinking, or think while I'm spelling, environment. This is where we get both absorption of nutrients and also secretion of waste. And so basically what happens is a cell gets too big, rate of transfer can't keep up with the needs. Okay, so now a thought question. So other than limits on cell size, why else would an organism consist of many small cells rather than fewer larger ones? And the main benefit is it allows us to have functional specialization. Which means that you can have some cells that are dedicated to specific processes, and it also allows redundancy. So that if you lose one cell, the whole organism doesn't go down. Okay, so now that we understand why cells are limited in their size, let's talk about the basic components of a cell. So this is an electron microscopic view of a cell. So if you start out here, this is the outer edge of the cell here. And so then we're going to describe what are the components that we're looking at. So starting with the outside, we have the plasma membrane, which is the outer membrane of the cell. Basically forming the border. We have the nucleus, which is membrane bound as well. And is a compartment that contains DNA. So it contains the DNA, which we talked about in the last lecture, is the information that tells the cell how to make all of its proteins, which basically tells the cell how to make itself. The third component of a cell is the cytosol. This also equals the cytoplasm. And this is a gel-like matrix that fills the cell. I don't know if you see that, but I just bumped something with my hand. So, gel-like matrix. Filling cell. This is also called the intracellular fluid. The last component of a cell, the last big broad category, are the organelles. And the organelles are membrane-bound compartments with specific functions. And so here's one organelle. You can see a variety of them scattered throughout the cell. Basically, it's specialized functions, which we'll be going into in a minute. Okay, so first part of the cell that I wanted to talk about is the cell surface, because this is the part of the cell that interacts with the environment. So as a way of review, so this is from the last lecture, what type of molecules are the membranes of cells made of? And so if you remember, these are lipids, and the specific type of lipid is a phospholipid. which you can see zoomed in down here. So it's a phospholipid bilayer, actually. So it's two sheets of phospholipids interacting to form this hydrophobic barrier. So 75% of the plasma membrane lipids are phospholipids. And again, phospholipids are created by this phosphoglycerol head. So that's up here, or also over here, which you can see contains a variety of oxygens, which makes this a hydrophilic component. And then fatty acids, which are these carbon chains coming down. This is a saturated one, so saturated, unsaturated, because it's got a kink in it, double bond. But fatty acids that also give the property of the phospholipid. So phospholipids are described as amphipathic. So amphipathic molecules have a hydrophilic region. So on a phospholipid, there's a hydrophilic head. And a hydrophobic tail. To remember the term amphipathic, so pathic means likes. Amphi means, I'm actually not sure what amphi means, but the way I remember it is amphibian, which amphibians live both in land and water. So amphi can be sort of like must mean something along the terms of sort of two different things, but likes both water and air. So water and no water. So it likes both water and no water. So this is a zoomed in vision of the phospholipid bilayer. So here's the bilayer here, and you can see you have a phosphate head with a fatty acid tail interacting, creating one sheet, and then a partner on the other side. And what this forms is a barrier between two aqueous environments or water environments. So aqueous just means water-filled environments. And then this region is going to be hydrophobic. So because you've got water, hydrophobic, and then water again, this forms a good barrier. When you look at the fatty acids or phospholipids in a bilayer, these phospholipids are constantly shifting around. So any one is kind of rotating. You could actually see them if you're looking at a sheet, basically looking at the head of one phospholipid, it will basically be migrating around that sheet. And this is called the fluid mosaic model. And so they're constantly moving laterally. Phospholipids move laterally in the membrane. They can occasionally flip-flop across the membrane, but this is a very rare event. So rarely shift sides. And what this allows is the cell is able to then have a phospholipid bilayer that has different properties on the outside versus the inside. So it allows differences between sides. So of the lipids that you find in plasma membranes, second most common is cholesterol. And the role of cholesterol in the plasma membrane is to regulate the fluidity. Meaning the more cholesterol, the more movement you get in the plasma membrane and the more flexible the membrane is. So you can see here cholesterol inserts itself in between the hydrophobic fatty acid tails of the plasma membrane, kind of spreading them out, and functions roughly the equivalent of having a unsaturated fatty acid tail. Second component of the plasma membrane are the proteins. And these proteins are called membrane-associated proteins. Here you can see the purple is a membrane protein that is embedded within the phospholipid bilayer. And even though these proteins are relatively rare, making up only 2% of the molecules, they make up about 50% of the total mass of a phospholipid bilayer, or plasma membrane. So what's a general property that you would expect to see in a protein that is associated with the phospholipid bilayer? So based on what you know about phospholipid bilayers, as there being amphipathic molecules, to integrate into the plasma membrane, protein must have both a hydrophobic and a hydrophilic region. So hydrophobic and hydrophilic. This allows the protein to then span both the hydrophobic region, but then have these components that stick out into the aqueous environment on either side that are hydrophilic. Because of this, the proteins would also be described as being amphipathic. There are two classes of membrane protein. There are the transmembrane or integral membrane proteins. So these ones over here, transmembrane or integral. And that's because they have this big component that spans the phospholipid bilayer, so allowing an interaction between the outside and the inside of the cell. The other class are the membrane-associated, or also called the membrane-bound proteins. And these proteins only span one side of the plasma membrane. So this one here is just anchored into the plasma membrane. This one actually has a part that's hydrophobic, integrating itself in. But the key thing being that they only interact with one side of the phospholipid bilayer. Proteins in the membrane have a variety of functions, just like proteins in the rest of the cell, where they have one of the more highly diverse molecules that we have. So proteins can be involved in transport, moving molecules, so these blue molecules from one side to the other. They can also be involved in signal transduction. So if you have a hormone or a neurotransmitter can come in, activate a receptor, and then send a signal into the cell to elicit a response. They're involved in cell-cell recognition, which you can see here. This would be sort of an immune response as well as location response, allowing the cell to know that it's in the proper location, or the immune system to think that that cell is not supposed to be there. They form anchors for cells. So this is an extracellular matrix attachment point, anchoring it into the extracellular matrix, allowing the cell to stay relatively put. It's a storage spot or anchor spot for enzymes that will function specifically at the plasma membrane, and then finally allows cells to stick together. So some of the membrane projections that you find that we'll talk about are microvilli. These are ones that are very important in the small intestine. So here you can see an individual villus sticking up. And what these do is increase the surface area. And when you increase the surface area, that means you get faster diffusion of molecules or transport of molecules across the membrane. So faster transmembrane membrane transport. These images are both electron microscope images. Here you can see these are actually tubules going down, anchoring, microtubules, anchoring the microvilli. And you can see the same thing over here. This is now a cross section, so if we did a cut like this, you're now looking at these in cross section, and you can see the tubules and then the plasma membrane around the outside. second structure that we're going to see in cells involved in a variety of physiology are our cilia. And cilia, unlike microvilli, are motile, so they aren't used to increase surface area, but include cytoskeletal elements that allow the cilia, little hairs on the surface of the cell, to move. So hairs on cell surface that are actively moved. And so you can see cytoskeletal elements again going up inside the cilia. Here's the electron microscope version of it. This is a cross-section of the individual cilia, where you can see it's got a very stereotypical pattern of microtubules and microfilaments. And then this is the diagram showing that same thing. But basically these tubules slide past each other, allowing the cilia to bend and move things. So the cilia basically are motor proteins that convert chemical energy into mechanical energy. So what we mean by that is ATP goes into movement. And so if the way that a cilia works is it's kind of like an oar. So if you see here's the power stroke, so you can see the cilia bends over, and then once it's bent all the way over, the recovery stroke kind of folds over on itself and then brings itself back upright. So the question is, why does it beat this way rather than just going back and forth? So going back and forth equally. And so the answer, so if it's equally stiff on both power and recovery strokes, just pausing to give you some time to think about that. So the reason, what that would do is basically accomplish nothing. So if the cilia doesn't bend over like it does here, then as it shifts back and forth, it's just going to push something back and forth and not really move it one direction. Whereas this, with a power stroke and recovery stroke, allows it to move substances across the surface of the cell. So move it one direction. The next membrane projection that we'll see over the year, and we won't see this till the very end of the year, or end of the course, are flagella. And flagella is a single projection here, sticking out of, this is a sperm cell, but it's a single projection rather than cilia, which are many, and it's used for swimming. So probably many of you have seen videos of sperm swimming around, and so they beat the cilia, they beat the flagella back and forth, and then that allows propulsion. The last of the membrane projections are the pseudopods, and they are basic pseudo for fake pods or feet, so these are fake feet, but a cell will reach out with these pseudopods, grab onto some extracellular matrix and pull itself, and then it's also used to engulf bacteria, which is what this purple cell is doing, or whatever color that is. Okay, last thing to talk about in cells are the organelles. And so again, organelles were those small specialized compartments within a cell. So here you can see the variety of organelles that you find in a cell. We're going to be talking through many of these, but not all of them. So this is the thing with the fuzz is a cell, and then here purple is the nucleus, but we'll go through all of that. So the way I like to approach thinking about these organelles, at least for a physiology course, is what are the functions that each organelle needs to do. So if we start out with things that need to be accomplished, a cell needs to be able to separate itself from the inside world to the outside world, needs to get rid of toxic waste, needs to detoxify any sort of toxin that it ingests or poisons that it ingests. It needs to generate energy, needs to store information on how to make new proteins and how to make new cells, and then finally it needs to take that information and build new parts. So first function, separating from the inside and outside world, is the plasma membrane, which will surround the cell and all of its organelles. Second function, dispose of waste. This is accomplished by the lysosomes. Lysosomes are cell compartments that contain digestive or hydrolytic enzymes. So hydrolytic enzyme compartments. And what they do is break down food particles and worn out parts. So peroxisome is an organelle that contains enzymes to oxidize organic molecules. And does this primarily by producing hydrogen peroxide. Produces H2O2, which is hydrogen peroxide. And what that will do is then inactivate or detoxify molecules. Activates, detoxifies. Or at least that's what it's evolved to do. This is not always the case. Sometimes the actual oxidation of a molecule can make it worse. But most of the time this is beneficial. Fourth function is to provide a constant supply of energy. This is accomplished primarily by the mitochondria, which doesn't do all of the glucose metabolism, but is where the efficient metabolism of glucose using oxygen occurs. So efficiently converts glucose energy to ATP. Fifth function is to store information. This is accomplished in the nucleus where DNA is stored. So it stores heritable info as DNA. Sixth thing is we need to take that information and build something with it. This is accomplished by ribosomes, which aren't actually organelles, they're just small proteins, or they're actually made up of nucleic acids themselves and some protein. But what they do is translate messenger RNA into polypeptide sequences. These ribosomes are located in a couple of different locations. So the first location are the cytosolic ribosomes, which are primarily responsible for synthesizing the proteins for the cell itself. Second location is on the rough endoplasmic reticulum. So here you can see the ribosomes are actually embedded in the plasma membrane of the rough endoplasmic reticulum. And what the rough endoplasmic reticulum does using these ribosomes is synthesize proteins that are either going to go to outside the cell, so proteins for secretion, or specific compartments within the cell. As part of building new parts, you also need to sort of tweak or modify these proteins, and this is accomplished by the Golgi apparatus, which then takes proteins that have been synthesized and modifies them. So it modifies proteins and then targets them to specific locations. targets to specific locations. Okay. So that can either be outside the cell or specific compartments. Another component of a cell that we haven't really talked about creating are the lipids, so like phospholipids and triglycerides and steroids, and these are synthesized by enzymes that are located within the smooth endoplasmic reticulum. So smooth endoplasmic reticulum contains enzymes to synthesize lipids. And with that, we are done with cell biology. So here's the list of terms that I think I added for today's lecture and the objectives I would like you to be able to do.